Consider this chemical reaction. One large compound is broken into two smaller compounds. This reaction takes place very slowly, much too slowly to be useful in a cell. In order to speed up chemical reactions, cells make a special class of proteins called enzymes. An area on the enzyme, called the active site, attracts the compound it will alter. This compound is called a substrate. Notice the shape of the substrate and the active site. They fit each other like a lock and key. In general, each enzyme's active site can fit only one kind of substrate, and so can speed up only one kind of reaction. The substrate attaches to the active site, forming the enzyme-substrate complex. A slight change in the shape of the substrate weakens a chemical bond. The weak bond is easily broken, splitting the substrate into two pieces. The pieces, now referred to as products, detach from the enzyme. In enzymatic reactions, only the substrate is changed. The enzyme itself is unaffected by the reaction. After the products leave the active site, the enzyme is free to catalyze another reaction. Enzymes cause reactions to occur quickly enough to be useful to a cell. A drop of red dye is added to a beaker of water. The color spreads as the dye molecules move randomly away from each other. In other words, there is a net movement of molecules from the area of high dye concentration, where the dye entered the water, to the area of low dye concentration, the water surrounding the dye. This process is called diffusion. With time, the dye becomes evenly distributed in the beaker. The water in the beaker is a single shade of pink, and diffusion no longer occurs. There is no net movement of dye from one area into another. Now think about another beaker of water. A membrane is added, dividing the beaker into equal compartments. The membrane is semi-permeable, so only water can pass through the membrane. Water moves from one compartment into the other. A solute is added to the water in both compartments, but not in equal quantities. More solute is added to the left side than to the right. The solute cannot pass through the membrane to even out the concentration, but water can. Water diffuses from the area of lower solute concentration, the right compartment, into the area of higher solute concentration, the left compartment, until the concentration of solute evens out. Diffusion through a semi-permeable membrane is called osmosis. The membrane surrounding a cell is semi-permeable. The cytoplasm of a cell is mostly water containing many dissolved solute molecules. Recall that water can flow freely across a semi-permeable membrane. Recall also that the movement of water across a semi-permeable membrane depends upon the concentration of solutes on either side of the membrane. Therefore, the movement of water into or out of a cell will depend on the relative concentration of solute inside versus outside the cell. A cell is placed in a hypertonic solution. The concentration of solutes in a hypertonic solution is higher than the concentration of solutes inside the cell. Water diffuses out of the cell. It shrivels and shrinks. Now think about a cell placed in a hypotonic solution. The concentration of solutes in a hypotonic solution is lower than the concentration inside the cell. Again, the flow of water acts to even out the solute concentration on either side of the cell membrane. Water diffuses into the cell, causing it to swell. The cell bursts as more and more water flows in. Finally, consider a cell placed in an isotonic solution. The concentration of solutes in an isotonic solution is equal to the concentration of solutes inside the cell. Although water flows in and out of the cell, there is no net flow in either direction. The cell neither shrinks nor swells. To stay alive, a cell must exchange materials such as food and wastes with its environment. Some materials are exchanged by a process called passive transport. Other materials must be carried across in a process called active transport. The membrane of a cell is semipermeable. Some small molecules, such as oxygen and water, diffuse freely across the cell membrane. Carbon dioxide and amino acids can also diffuse into or out of the cell. The net flow of molecules depends upon their concentration inside versus outside the cell. Molecules tend to move from an area of high concentration to an area of low concentration. No energy is required to move the molecules. This is an example of passive transport. Molecules with strong electrical charges, such as ions, cannot diffuse across the membrane. Although the ions may be small, their charge prevents them from moving through the layer of lipid tails in the middle of the cell membrane. Many molecules, including proteins, are simply too large to diffuse across the cell membrane. Carbohydrates, such as sugars and starches, are also too large. Large molecules are transported across the membrane by carrier proteins in the membrane. Glucose, a sugar, is one molecule transported in this way. Glucose attaches to the carrier protein. The protein changes shape and releases the glucose molecule on the opposite side of the membrane. Glucose diffuses down its concentration gradient, from the area of higher concentration to the area of lower concentration. This process, called facilitated transport, requires no energy. It is therefore another example of passive transport. Cells often pump molecules against a concentration gradient. Ions, such as sodium and potassium, are moved in this way. Again, a special carrier protein, called the sodium-potassium pump, is involved. ATP transfers energy to the sodium-potassium pump. The protein uses this energy to change its shape. Sodium ions bind to the carrier protein. Again, the shape of the protein changes. The sodium ions are released outside the cell. The protein can now bind potassium ions. When the potassium ions bind, the protein changes shape. The potassium ions are released inside the cell. The energy gained from ATP has been used up. The sodium-potassium pump needs energy from another ATP to repeat the cycle. Sodium ions are moved against their concentration gradient. Potassium ions are also moved against their concentration gradient. Since this process requires energy, it is an example of active transport. Cells can move large particles across the cell membrane by bulk transport. Bulk transport into the cell is called endocytosis. Bulk transport out of the cell is called exocytosis. During endocytosis, the cell membrane folds into a pouch surrounding particles outside the cell. When the particles are completely enclosed, the pouch closes off. A vesicle is formed. This vesicle carries the particles into the cell and can release its contents into the cytoplasm. Human white blood cells engulf bacteria and other unwanted cells. This process is called phagocytosis. Phagocytosis is another example of endocytosis. Once inside the cell, the vesicle containing the bacteria fuses with another vesicle called a lysosome. The lysosome contains digestive juices which destroy the bacteria. Exocytosis is the opposite of endocytosis. Particles are transported out of the cell. A vesicle containing wastes, or cell products, moves toward the cell membrane. The membrane surrounding the vesicle fuses with the cell membrane. The contents of the vesicle are secreted as the membrane smooths out. The molecule used by most organisms to transfer energy is called adenosine triphosphate, or ATP. An ATP molecule has three parts: adenine, ribose, and phosphate groups. Adenine is one of the bases found in DNA and RNA. Ribose is a sugar molecule found in RNA. The adenine and ribose portion of the ATP molecule is called adenosine. The three phosphates linked to the ribose in a chain are called triphosphate. Adenosine together with the three phosphate groups is called adenosine triphosphate, or ATP. Notice the wavy lines between the two outer phosphates. These wavy lines represent high-energy bonds. Breaking the first high-energy bond in ATP releases energy and a free phosphate. This energy is used to power cellular activities. Adenosine, with only two attached phosphate groups, is called adenosine diphosphate, or ADP. Energy from the breakdown of food is used to attach a third phosphate group to ADP. A high-energy bond is formed between the second and third phosphate groups, resulting in an ATP molecule. Energy from food is used to change the ADP to an ATP. The energy stored in ATP is transferred and released when needed to power cellular activities. This process may occur as many as 10 million times per second in some cells. Plants convert light energy into a form usable by cells. This process is called photosynthesis. Leaves are the primary light-collecting organs of plants. The cells in leaves contain specialized organelles called chloroplasts. Photosynthesis occurs within the chloroplast. Each chloroplast consists of two membranes, the outer membrane and the inner membrane. Fused layers of inner membrane form disc-shaped structures called thylakoids. A stack of thylakoids is called a granum. The grana are surrounded by a gel-like matrix called the stroma. Thylakoids convert light energy into the chemical energy of ATP. Each thylakoid is a closed compartment. The space within is called the thylakoid space, or lumen. Groups of light-collecting molecules called photosystem II and photosystem I are embedded within the thylakoid membrane. Proteins called electron carriers serve as messengers between these photosystems. An additional protein, the ATP synthase, produces ATP. Light-striking photosystem II is absorbed, and its energy is used to split water into one atom of oxygen, two hydrogen ions, and two electrons. The hydrogen ions stay in the thylakoid space while the electrons move into photosystem II. Light energizes the electrons. They are then accepted by the first protein in the series of electron carriers. This protein carries them to the second carrier. Energy from the electrons is used to pump hydrogen ions across the thylakoid membrane. As a result, the concentration of hydrogen ions in the thylakoid space increases. The electrons are accepted by the third electron carrier. This carrier delivers them to photosystem I. Light energizes the electrons again. The energized electrons move to the final carrier protein, which adds them to NADP+ to form NADPH. Recall that the concentration of hydrogen ions is greater inside the thylakoid space than outside in the stroma. Therefore, hydrogen ions readily diffuse out of the thylakoid. The protein ATP synthase uses the energy released by this flow to convert ADP to ATP by the addition of a phosphate. The chemical energy stored in ATP can then be used by the cell to power cell functions. The light-dependent reactions of photosynthesis take place in the thylakoids. Light energy powers the formation of two types of high-energy compounds. One compound is NADPH, and the other is ATP. The plant cell uses the energy in these compounds in the second phase of photosynthesis called the Calvin cycle. The Calvin cycle takes place in the stroma surrounding the thylakoids. Each turn of the Calvin cycle builds one small subunit that leaves the loop. These small subunits are the building blocks for sugars and starches. The starting compound for the Calvin cycle is a 5-carbon compound called RUBP. During the first step of the cycle, one molecule of carbon dioxide is added to each of 3 RUBP molecules. The plant absorbs carbon dioxide from the surrounding air. The 3 RUBPs become 6 carbon molecules. Each of these molecules splits in half, forming 6 3-carbon molecules called PGA. 6 ATPs must be spent to energize the 6 PGAs. Then 6 NADPHs must be spent to energize the PGAs still further. These 3-carbon molecules are now called PGAL. Of the 6 PGALs formed, 5 remain in the Calvin cycle while 1 exits. As the cycle continues, the 5 PGALs rearrange back into 3 molecules of RUBP, each with 5 carbons. This requires 3 more ATPs. Each time the Calvin cycle turns, the 5-carbon RUBPs accept carbon dioxide molecules. 6 new PGALs form at the cost of ATPs and NADPHs. A PGAL molecule leaves the cycle, and the 5-carbon starting material is regenerated once again. The PGAL that left the Calvin cycle moves out of the chloroplast and enters the plant cell's cytoplasm. Once in the cytoplasm, the PGAL can be combined with another PGAL into a 6-carbon glucose molecule, or other sugar. All organisms are made up of living cells, and all living cells need energy to power life processes. When a cell needs energy, a glucose molecule is broken down into CO2 in a long series of reactions. The first series of reactions is called glycolysis. The cell must invest 2 molecules of ATP at the start of glycolysis. This investment allows the 6-carbon glucose to split into 2 energized molecules containing 3 carbons each. Energy is harvested. All organisms are made up of living cells, and all living cells need energy to power life processes. When a cell needs energy, a glucose molecule is broken down into CO2 in a long series of reactions. The first series of reactions is called glycolysis. The cell must invest 2 molecules of ATP at the start of glycolysis. This investment allows the 6-carbon glucose to split into 2 energized molecules containing 3 carbons each. Energy is harvested from the 3-carbon compounds in a series of small steps. Each 3-carbon compound loses 2 high-energy electrons, and these combine with the compound NAD+, forming the higher energy compound NADH. Next, energy from the 3 carbon compounds is transferred to ADP, and 2 ATPs are formed. A bit more energy is transferred from the 3 carbon compounds to ADP. 2 more high-energy ATPs are formed. This is the last reaction in glycolysis. The 2 3-carbon products are pyruvate molecules. Although the glycolysis of one glucose molecule costs the cell 2 ATPs initially, there is still a net gain of 2 NADHs and 2 ATPs for each glucose broken down. The 3-carbon pyruvate molecules formed during glycolysis still contain most of their chemical energy. This energy is harvested in a series of reactions called aerobic respiration. Aerobic respiration takes place inside organelles called mitochondria. The pyruvate molecule passes through the outer membrane of the mitochondrion and into the outer compartment. The pyruvate molecule then passes through the inner membrane and enters the mitochondrial matrix. The first series of chemical reactions in aerobic respiration takes place in the mitochondrial matrix. The pyruvate molecule is split and enters a continuous loop of reactions. This loop of reactions is called the Krebs cycle. As these reactions take place, energy from the carbon compounds is used to make NADH, ATP, and FADH2, all high-energy compounds. Carbon dioxide is also formed and released. These carbon dioxide molecules leave your body when you exhale. Before entering the Krebs cycle, another pyruvate molecule is split. Carbon dioxide is released. Next, 2 electrons are transferred from the 2-carbon compound to NAD+. NADH is formed. The 2-carbon compound becomes associated with a molecule called coenzyme A. Scientists call the resulting molecule acetyl-CoA. Acetyl-CoA enters the Krebs cycle, and the acetyl portion becomes linked with a 4-carbon compound. The result is a 6-carbon molecule called citric acid, the first intermediate in the Krebs cycle. 1 carbon is removed, taking with it 2 oxygen atoms. The resulting molecule, carbon dioxide, is released. The 5-carbon molecule continues through the cycle. 2 electrons are transferred to an NAD+ molecule. NADH is formed. A similar sequence of events takes place again. This time, the 5-carbon compound is changed to a 4-carbon compound. Another carbon dioxide forms and exits. Another pair of electrons is transferred to NAD+, producing NADH. The leftover 4-carbon compound is still energized. It gives up a bit of its energy to an ADP molecule, and ATP forms. A pair of electrons is transferred to a different kind of electron carrier called FAD. The higher energy FADH2 forms. A new reaction results in a slightly different 4-carbon compound. One last reaction of the Krebs cycle liberates 2 more electrons from the 4-carbon compound, and they convert another NAD+ to NADH. The remaining de-energized 4-carbon compound is the starting material for a new round of the Krebs cycle. Recall that a glucose molecule is split into 2 3-carbon pyruvate molecules during glycolysis. Therefore, the chemical reactions in the Krebs cycle occur twice for each glucose molecule that enters the glycolysis pathway. This is a substantial harvest of high-energy compounds. the high-energy compounds made during the Krebs cycle remain in the mitochondrion and power a second phase of aerobic respiration, the electron transport chain. Most of the energy a cell harvests during the Krebs cycle is held in energy carriers like NADH and FADH2. These energy carriers stay within the matrix of the mitochondrion. NADH and FADH2 transfer their high-energy electrons to proteins on the inner membrane of the mitochondrion. The inner mitochondrial membrane separates the matrix from the outer compartment. Large proteins are embedded in the inner membrane. These proteins are the electron carriers in the electron transport chain. The electron carriers are arrayed side by side like the links in a chain. 3 of these proteins are ion pumps. NADH transfers 2 high-energy electrons to the first electron carrier, an ion pump. NADH changes to NAD+. The carrier protein uses energy from the electrons to pump 2 hydrogen ions from the matrix into the outer compartment. The electrons are then passed to the next protein in the chain, and then on to the third electron carrier. This third protein is also an ion pump. It uses a bit more of the electron's energy to pump 2 hydrogen ions through the membrane. The electrons shift again to the fourth protein and then on to the fifth protein. This protein is a third ion pump. Using energy from the electrons, the protein pumps 2 more hydrogen ions into the outer compartment. The 2 electrons are now much lower in energy. They combine with 2 hydrogen ions and an oxygen atom. Oxygen, absorbed from the air when you inhale, is the final electron acceptor at the end of the electron transport chain. A molecule of water, H2O, is formed. A similar sequence of events takes place with FADH2. FADH2 transfers electrons to the second ion pump in the chain. The FADH2 is converted to FAD. Hydrogen ions are pumped through the protein into the outer compartment. The electrons shift down to the last protein in the chain. More hydrogen ions are pumped. Again, oxygen serves as the final electron acceptor, and a water molecule forms. Ion pumps are powered by energy from electrons. The ion pumps move hydrogen ions from the matrix into the outer compartment. Hydrogen ions concentrated in the outer compartment represent a form of potential energy, like water held behind a dam. They diffuse back into the mitochondrial matrix through a special protein called ATP synthase. The diffusion of hydrogen ions across the membrane energizes the ATP synthase. This energy is transferred to an ADP molecule, causing an ATP to form. The formation of ATP provides the cell with usable fuel in large amounts. The cell cycle includes all phases of a cell's life. For most of the cell cycle, the cell is in interphase. During this phase, the cell grows and matures. The chromosomes of DNA exist as long, thin strands within the nuclear envelope and nucleolus. 2 centrioles, a single pair, lay at right angles to each other just outside the nucleus. Near the end of interphase, the cell prepares to divide. The DNA is replicated at this time, producing identical strands of DNA called sister chromatids. The sister chromatids are attached to each other at the centromere. The centrioles are also duplicated. The division of the cell nucleus into two identical daughter nuclei now begins. This process is called mitosis. Prophase is the first phase of mitosis. During prophase, the replicated chromosomes condense. The sister chromatids stay attached at the centromere. The two centriole pairs move to opposite sides of the cell. The nucleolus and the nuclear envelope dissolve. The mitotic spindle forms between the two centriole pairs. A spindle fiber attaches to the centromere on each chromosome. The spindle fibers begin to move the chromosomes to the center of the cell. The second phase of mitosis is metaphase. The chromosomes line up on a plane in the center of the mitotic spindle. The third phase of mitosis is anaphase. The centromere of each chromosome divides. The sister chromatids separate and are pulled toward opposite ends of the cell by the spindle fibers. The fourth and final stage of mitosis is telophase. Daughter nuclei form at each end of the cell. Mitosis is then complete, but the process of cell division continues. During cytokinesis, the cell membrane begins to pinch the cytoplasm of the cell in half. Cytokinesis and the process of cell division is complete when two new daughter cells are formed. After cell division, the cells re-enter interphase and begin another cell cycle. The chromosomes uncoil and the cell resumes normal activity. The process of meiosis results in 4 daughter cells that have half the number of chromosomes found in the parent cell. In this case, the parent cell has 4 chromosomes, while the daughter cells have only 2 chromosomes. The chromosomes are shown here as they look during cell division. In the parent cell, the chromosomes have replicated and condensed. Each chromosome consists of 2 sister chromatids joined at the centromere. The parent cell has 2 sets of chromosomes. One set of chromosomes came from the mother, here colored in red. The other set came from the father, here colored in blue. The matching pairs of chromosomes, one red and one blue, are called homologous chromosomes. Homologous chromosomes carry genes for the same traits and are the same size and shape. A cell that contains 2 complete sets of chromosomes is called a diploid cell. The parent cell divides twice during meiosis. The first cell division is called meiosis 1. The matching pairs of chromosomes, or homologous chromosomes, separate during meiosis 1. The 2 newly formed cells therefore contain only half the chromosomes of the parent cell. A cell with only one set of chromosomes is called a haploid cell. The second cell division is called meiosis 2. The sister chromatids of each chromosome separate during meiosis 2. The 4 daughter cells are haploid cells. Imagine a diploid cell during interphase. The chromosomes are long, thin strands of DNA within the nuclear envelope. There is a single pair of centrioles near the nucleus. Near the end of interphase, the cell prepares for the first phase of meiosis, meiosis 1. The pair of centrioles duplicates. Each chromosome is replicated. Homologous chromosomes begin to group together. Meiosis 1 begins. The first phase of meiosis 1 is prophase 1. During prophase 1, the centriole pairs move to opposite sides of the cell. The chromosomes condense. The homologous chromosomes are tangled together. A total of 4 chromatids are present, since each chromosome was replicated before meiosis began. Each pair of sister chromatids is joined at a centromere. While the pairs of chromosomes are close together, they may break and exchange genes. This is called crossing over. The nuclear envelope dissolves and the spindle fibers form. The homologous chromosomes stay in pairs as the spindle fibers pull them toward the center of the cell. The second phase of meiosis I is metaphase I. During metaphase I, the homologous chromosomes line up in the center of the spindle. The third phase of meiosis I is anaphase I. During anaphase I, the homologous chromosomes are pulled apart by the spindle fibers. They move to opposite ends of the cell. The fourth phase of meiosis I is telophase I. The nuclear envelope forms around the new sets of chromosomes. Cytokinesis begins when the cell membrane starts to pinch the cell's cytoplasm in half. 2 new daughter cells are formed. Each daughter cell is haploid. It has half of the chromosomes found in the parent cell. In preparation for another cell division, the centriole pairs duplicate. The chromosomes do not replicate. Each daughter cell is now ready for meiosis II, the second cell division of meiosis. Meiosis II is very similar to mitosis. However, meiosis II starts with a haploid cell. The first phase of meiosis II is prophase II. During prophase II, the centriole pairs move to opposite ends of the cell. The nuclear envelope dissolves, a spindle forms, the chromosomes are pulled toward the middle of the cell. During metaphase II, the second phase of meiosis II, the chromosomes line up in the center of the spindle. In anaphase II, the third phase of meiosis II, the centromeres of the sister chromatids separate, and the sister chromatids of each pair move toward opposite ends of the cell. Each sister chromatid is now an individual daughter chromosome. The last phase of meiosis II is telophase II. During telophase II, nuclear envelopes form around the chromosomes at the end of the cell. In cytokinesis, the cell membrane pinches the cell in half. Each haploid cell has now divided into 2 haploid daughter cells. From the original cell, there are now 4 new daughter cells, each with 1 set of single chromosomes. The chromosomes of all organisms contain regions called genes. Each gene codes for a characteristic of the organism. For example, a specific gene in pea plants codes for seed color. Another gene codes for seed shape. A diploid cell has 2 sets of chromosomes. Matching pairs of chromosomes are called homologous chromosomes. Homologous chromosomes carry the same genes in the same locations. Since all diploid cells have homologous chromosomes, the cell carries 2 copies of each gene. These 2 copies may not be identical, however. The genes may code for different versions of the same trait, seed color, for example. One version of the gene on the chromosome may code for green seeds, while the version on the other chromosome may code for yellow seeds. The different versions of a gene are called alleles. The cell also carries 2 different alleles for seed shape. One allele codes for wrinkled seeds, and the other allele codes for smooth seeds. The cell prepares for meiosis. The chromosomes replicate. In metaphase I of meiosis, homologous pairs of chromosomes move to the middle of the cell. In anaphase I, the homologous chromosomes separate from each other and are pulled to opposite ends of the cell. Two daughter cells form at the end of meiosis I. When the homologous chromosomes separated, the alleles on the chromosomes separated as well. The allele for yellow seeds is in the right daughter cell, while the allele for green seeds is in the left daughter cell. This process is called segregation. The alleles for seed shape have also segregated. The allele for wrinkled seeds is in the left daughter cell, while the allele for smooth seeds is in the right daughter cell. During prophase I of meiosis, homologous chromosomes are grouped together. The chromosomes are sometimes broken when their arms overlap. When the breaks are repaired, the pieces of the two chromosomes may be exchanged. This process is called crossing over. Due to crossing over, one chromosome can exchange alleles with another. After crossing over, each chromosome in the homologous pair has a different combination of genes than it had before meiosis began. Recall that crossing over is the exchange of genes between homologous chromosomes. During prophase I of meiosis, homologous chromosomes are grouped together. This is when crossing over can occur. If the arms of the chromosomes line up unevenly, however, errors may occur in the crossing over process. Homologous chromosomes may be broken in different places. This produces pieces of different sizes. The longer piece contains a gene that the shorter piece is lacking. When the breaks are repaired, the different sized pieces may be exchanged. When the short piece of the chromosome is added to the shortest broken arm, a gene is left out. The gene represented by the blue band is missing. This error is called a deletion. The longer piece contains genes already present on the other chromosome's arm. When the long piece is attached, the chromosome gains a second copy of a gene. This error is called a duplication. When the pairs segregate and the chromatids separate in meiosis II, 4 chromosomes are produced. 2 of them have new combinations of genes. The arrangement and number of genes within a chromosome can be changed by errors called translocations and inversions. Non-homologous chromosomes may line up next to one another. A piece of one chromosome may break off and become attached to the other nearby chromosome. As a result, one chromosome gains genes that are lost by the other. This kind of transfer is called a translocation. The genes on a single chromosome can also be rearranged. A fragment of a chromosome may break away, turn completely around, and reattach itself in reverse order. This rearrangement is called an inversion. Sometimes homologous pairs of chromosomes do not separate correctly during meiosis. This leads to daughter cells with an abnormal number of chromosomes. Meiosis I may proceed normally. Homologous pairs of chromosomes line up in the center of the cell and separate, sending one member of each pair to opposite ends of the cell. The cell divides, forming normal haploid daughter cells. Meiosis II also begins normally for the 2 daughter cells. But during anaphase II, in one of the cells, 2 sister chromatids fail to separate. Both chromatids are pulled toward the same end of the cell. When these cells form gametes, one gamete will have 2 copies of the same chromosome. The other gamete will not get a copy of that chromosome at all. An error of this type is called a non-disjunction. The 2 gametes produced by non-disjunction have an abnormal number of chromosomes. Deoxyribonucleic acid, or DNA, is a molecule made up of two long strands. The strands wrap around each other, forming a double helix. The DNA double helix resembles a twisted ladder. The sides of the ladder are formed by alternating phosphate and sugar groups. The sugar and phosphate groups form the backbone of a strand of DNA. The sugar in the DNA backbone is called deoxyribose. The two backbones in a DNA molecule face in opposite directions. Pairs of bases form the rungs of the DNA ladder. Hydrogen bonds hold the pairs of bases together. There are 4 different bases in DNA: thymine, adenine, cytosine, and guanine. The names of the bases are often abbreviated. T stands for thymine, A for adenine, C for cytosine, and G for guanine. T always pairs with A, C always pairs with G. A TA pair and a CG pair are called complementary base pairs. Each group of phosphate, sugar, and base is called a nucleotide. DNA copies itself through a process called replication. At the beginning of replication, the double helix unwinds. Enzymes break the hydrogen bonds holding the base pairs together, and the strands separate. Each strand of DNA is copied by an enzyme called DNA polymerase. Each old strand of DNA serves as a template for a new complementary strand. The new strands are made from free-floating nucleotides by DNA polymerase. Complementary base pairs are matched. A always pairs with T, and G always pairs with C. As each new nucleotide is added to the chain, covalent bonds are formed between the sugars and phosphates along the new backbone. One new DNA strand is continually assembled base after base from its end. However, the other strand is built in the opposite direction in short fragments. One strand is made in fragments because DNA polymerase can work in only one direction. Because the two backbones in a DNA molecule face in opposite directions, the enzyme must work backwards to assemble one strand. After 2 fragments have been built, a covalent bond is formed between them. The process of replication continues. A's pair up with T's, and C's pair up with G's. Until 2 identical double helices have been formed. Instructions for making proteins are coded on segments of DNA called genes. But proteins are not made in the nucleus. A strand of ribonucleic acid, or RNA, carries the instructions for making proteins out of the nucleus. This type of RNA is called messenger RNA. Both DNA and RNA are long chains of nucleotides. Each nucleotide contains a sugar, a phosphate group, and one of 4 bases. RNA is different from DNA in 3 important ways. First, RNA is a single strand. Second, RNA has a different type of sugar, called ribose, in its nucleotides. Third, RNA contains the base uracil instead of thymine. The other bases found in RNA are the same as in DNA. They are cytosine, guanine, and adenine. The manufacture of RNA from a DNA template is called transcription. Transcription begins when the 2 strands of DNA in a specific region unwind and separate. Only one strand of the DNA serves as a template for the new RNA. The enzyme RNA polymerase matches RNA nucleotides with the complementary ones on the template strand of DNA. Recall that RNA contains uracil instead of thymine. Therefore, uracil pairs with adenine on the DNA strand. Transcription continues. A pairs with T on the DNA strand. C pairs with G, and U pairs with A. As the nucleotides are assembled in the proper sequence, RNA polymerase moves along the DNA strand, assembling the strand of RNA until the messenger RNA is completed. Hydrogen bonds are formed again between the complementary bases in the DNA molecule, and the DNA coils back up. The coded instructions on the DNA molecule have been copied to the newly manufactured strand of messenger RNA. A protein is a chain of amino acids. The process of linking amino acids together to make a protein is called protein synthesis. Protein synthesis occurs in the cytoplasm of a cell. mRNA leaves the nucleus through a nuclear pore after it is transcribed from DNA. The assembly of protein takes place on ribosomes in the cytoplasm. Ribosomes consist of two subunits, the small ribosomal subunit and the large ribosomal subunit. Floating within the cytoplasm are amino acids and transfer RNAs, or tRNAs. Each kind of amino acid binds to its own specific type of tRNA. Notice the 3-base region on the tRNA. It is complementary to a 3-base region on the mRNA. Each group of 3 bases on the mRNA is called a codon. The complementary 3-base sequence on the tRNA is called the anticodon. A small ribosomal subunit binds to the mRNA at the start codon, AUG. A specific tRNA carrying the amino acid methionine binds to the mRNA. The anticodon on the tRNA matches the codon on the mRNA. The large ribosomal subunit now binds to the small ribosomal subunit and the mRNA. A second tRNA with its attached amino acid binds to the second codon of the mRNA. The first amino acid is transferred from its tRNA to the second amino acid, and a bond between the two amino acids is formed. The protein chain has begun to grow. The first tRNA detaches from the mRNA. The ribosome moves down the mRNA to the third codon. This codon is recognized by the anticodon of another tRNA carrying an amino acid. The growing protein chain is transferred to the third amino acid. The second tRNA detaches. The ribosome again moves along the mRNA. A codon is exposed. Again, the codon is matched by the anticodon of a tRNA carrying an amino acid. The protein chain is shifted, adding another amino acid to the chain. The tRNA that lost its amino acid detaches, and the ribosome moves down the mRNA strand to the next codon. This process, called elongation, builds proteins one amino acid at a time. The ribosome continues to move along the mRNA. The 3 bases that make up a codon determine which tRNA attaches. Since each tRNA carries a specific amino acid, the sequence of amino acids in the new protein is determined by the order of bases in the mRNA. At the end of the mRNA, the ribosome encounters a stop codon, such as UGA. The completed protein is released. The tRNA floats off, and the mRNA detaches from the ribosome. The ribosome splits into small and large subunits. Human insulin can be produced by transferring the insulin gene from human DNA to bacterial DNA. The human gene for insulin is bordered on each side by a specific sequence of bases, G, A, A, T, T, C. A special enzyme added by a scientist recognizes this sequence and cuts the DNA on either side of the insulin gene. This isolates the insulin gene and leaves two segments of bases on either side. These segments are called sticky ends because they will bind to DNA segments that have complementary bases. The insulin gene is now ready for insertion into bacterial DNA. Bacteria, carrying the gene for tetracycline resistance, are used to hold the insulin gene. Resistant bacteria are needed for two reasons. First, this gene is found on plasmid DNA. Plasmids are circles of DNA separate from the main strand of bacterial DNA. Second, the tetracycline resistance gene is known to contain the GAATTC sequence of bases. The plasmid DNA is removed from the bacterium and treated with the enzyme used to isolate the human insulin gene. The enzyme cuts the plasmid DNA at the GAATTC sequence. The two ends are sticky, just like the ends on the human insulin gene. The insulin gene with sticky ends is mixed with the cut plasmid DNA. Complementary bases pair up. With the help of the enzyme DNA ligase, the DNA backbones are joined. The human insulin gene is now part of the bacterial DNA plasmid. The plasmid is then reinserted into a bacterial cell. Cells containing the insulin gene can be identified because they are no longer resistant to tetracycline. The resistance gene is cut and no longer functions. The bacterial cell grows and divides. Each new bacterial cell produces human insulin from the human insulin gene carried on the plasmid. The insulin is purified and used to treat people whose bodies cannot make their own insulin. Viruses consist of a core of nucleic acid surrounded by a protein coat. A virus cannot reproduce independently. In order to reproduce, it must infect a living cell. The infected cell is called the host. The process of reproduction for viruses is called viral replication. In the first stage of viral replication, the virus attaches itself to the host and injects its nucleic acid. In this case, the viral nucleic acid is DNA. Inside the host, the DNA forms a circle. Viral replication continues through one of two cycles, the lytic cycle or the lysogenic cycle. In the lysogenic cycle, the virus does not immediately kill the host. Instead, the viral DNA is incorporated into the host cell's DNA. The cell is now called a lysogenic host cell. The incorporated viral DNA is called a prophage. The viral DNA is dormant in the prophage form. The lysogenic host cell reproduces normally, replicating both its own DNA and the prophage. At any time, the prophage may separate from the host's DNA. When the viral DNA is free in the host cell, it can enter the lytic cycle. In the lytic cycle, viral replication rapidly kills the host cell. The virus takes control of the host. The host cell is forced to make copies of the virus by replicating the viral DNA and producing viral proteins. Thousands of copies of the virus are assembled within the host cell. When they are completed, the cell bursts open and the viruses disperse. The new viruses are able to infect other cells. A skeletal muscle causes motion by contracting. During contraction, the muscle thickens and shortens. When the muscle relaxes, it returns to its normal shape and length. Muscles contain many bundles. Within each bundle are many long muscle cells. These cells are called muscle fibers. Unlike most body cells, muscle fibers have many nuclei. Each muscle fiber has a pattern of light and dark bands called striations. The thin dark lines in the middle of the light bands are called Z-lines. The pattern of bands and Z-lines runs the length of each muscle fiber. Inside the muscle fiber are several myofibrils. The myofibrils contain muscle filaments. It is these filaments within the muscle fiber that give the cell its banded appearance. There are two types of filaments within a myofibril: thick filaments and thin filaments. The Z-lines are actually disc-like sheets of protein to which the thin filaments are attached. The segment of the myofibril between two Z-lines is called a sarcomere. The thin filaments are constructed of a protein called actin. The thick filaments are bundles of protein molecules called myosin. Each myosin molecule has a tail region and head region. The tails of the myosin molecules clump together while the heads protrude outward. Some of the myosin heads are attached to actin molecules forming cross bridges. The myosin molecules are oriented with their heads toward the ends of the thick filament. As a muscle begins to contract, it shortens and thickens. Now think about the sarcomere. When the muscle is relaxed, the free ends of the thin filaments are far apart. The Z-lines are also far apart. As the muscle contracts, the thin filaments are pulled toward the middle of the sarcomere until the thin filaments almost completely overlap the thick filaments. The Z-lines are pulled together, shortening the sarcomere. As a result, the muscle shortens. When the muscle relaxes, the sarcomere lengthens again. Muscle contraction can be seen in more detail by looking at the interaction between thick and thin filaments. Muscle contraction begins. ATP binds to a myosin head on the filament. As the ATP is broken down, the myosin head rotates away from the thin filament, releasing the cross bridge. The myosin head then rotates back toward the thin filament and makes contact with the next actin molecule. When contact is made, the ADP molecule is released, and the myosin head swivels forcefully, pulling the thin filament. The myosin cross bridge is now ready to start another cycle. As many myosin heads bind, swivel, and release, the thin filaments are pulled along the thick filaments. The thin filaments overlap the thick filaments more and more, shortening the sarcomere. When muscle contraction stops, the myosin cross bridges do not bind to the thin filaments. The thin filaments slide back to their original position, and the sarcomere lengthens. The muscle is now relaxed. All neurons, or nerve cells, have a cell body, an axon, and one or more dendrites. Dendrites receive signals from other cells and carry them to the cell body. The axon carries signals away from the cell body and toward other cells. The nervous system carries signals through the body in the form of nerve impulses. A nerve impulse is a wave of chemical and electrical change that moves along the membrane of the nerve cell. Between impulses, the membrane is in its resting state. In the resting state, the inner surface of the nerve cell membrane has a negative charge compared to the outer surface. The outer surface has a positive charge. This difference in electrical charge across the membrane is called the resting potential. When the nerve is stimulated, local channels in the membrane open, and sodium ions flow into the cell. The flow of these positively charged ions reverses the electrical charge across the membrane. The reversal of charge in this region of the membrane causes channels in the neighboring region to open. Sodium ions flow in, and the charge is now reversed in this region. A few thousandths of a second after the sodium ions enter the cell, other membrane channels open and allow potassium ions to flow out of the cell. This outflow of positive ions restores the membrane to its resting potential. This reversal of charge, followed by the restoration of the resting state, is called an action potential. Action potential is another name for nerve impulse. Action potentials travel very rapidly along the membrane of a nerve cell. Some move at a speed of 300 feet per second. The place where a nerve impulse is passed from a neuron to another cell is called a synapse. A synapse is a tiny gap between two cells. When a nerve impulse reaches the end of the axon, information is sent across the gap in the form of a chemical messenger. The chemical is called a neurotransmitter. Molecules of neurotransmitter are stored in small sacs at the end of the axon. On the other side of the synapse, receptors line the surface of the next cell. The arrival of a nerve impulse makes some of the sacs fuse with the axon's membrane. Each fused sac releases its neurotransmitter molecules into the synapse. The neurotransmitter molecules diffuse across the gap and bind to the receptors on the cell membrane. The binding of the neurotransmitter to the receptors causes a change in the membrane potential. This change triggers a nerve impulse. The neurotransmitters are then released by the receptors. They are either broken down or reabsorbed by the first cell. The synapse is ready to transmit another impulse. A virus or organism that causes an infectious disease is called a pathogen. The body's immune system recognizes a pathogen as foreign because of molecules on the pathogen's surface. These molecules are called antigens. When pathogens enter the body, they encounter antibodies. Antibodies are Y-shaped proteins with sites on their tips that bind to specific antigens. The body makes many different kinds of antibodies. Each antibody will bind only to a specific antigen. Because each antibody has two binding sites, clumps of antigens and antibodies can begin to form. Clumped antigens are not active, and the pathogens are not able to infect other cells. Macrophages recognize these clumps of pathogens and engulf them. The pathogens are then destroyed within the macrophage. Cell-mediated immunity is one way the body fights an infection and cancer. Two types of cells involved in cell-mediated immunity are macrophages, which engulf and destroy pathogens, and T cells. There are three types of T cells: helper T cells, killer T cells, and suppressor T cells. Pathogens are disease-causing organisms. Pathogens have unique molecules on their cell surfaces called antigens. These body cells are about to be infected by a pathogen. The pathogen enters the cell, where it disassembles. Antigens appear on the surface of the infected cell. Some of the pathogens are attacked directly by the body's macrophages before they can invade a cell. Once a pathogen is destroyed, pieces of the antigens appear on the surface of the macrophage. The rest of the pathogen is destroyed by the macrophage. A site on the surface of a helper T cell recognizes the antigen on the macrophage. Once the helper T cell binds to the macrophage at its recognition site, the macrophage sends a chemical signal to the helper T cell. The helper T cell is stimulated to divide many times, producing clones of identical helper T cells. Each helper T cell has the antigen recognition site of its parent. The helper T cells secrete a chemical substance that activates killer T cells. The activated killer T cells recognize the antigen on the surface of the infected cell and bind to it. The killer T cell releases a substance called perforin that makes holes in the membrane of the infected cell. The cytoplasm of the cell erupts through these openings. The cell is destroyed. The killer T cell moves to another cell, and the process of recognizing the antigen, binding, and destroying the cell is repeated. When antigens are no longer present, the suppressor T cells turn off the immune response by releasing a chemical that suppresses the activity of other T cells.